Hi everyone. Today we are going to talk about comparison of lipid accumulation in yeast and filamentous fungi. Oils and fats classed as lipids are substances of vegetable and animal origin that are widely found in nature and form the third major group of macronutrients as proteins and carbohydrates. Lipids do occur in cells of high plants, animals, fungi, and microorganisms. They contribute to cellular structure as being the major components of membranes, participate in biological reactions, protect the body against cold and other environmental influences, and act as energy storage molecules besides their role in human nutrition. One of their important physiological roles is that they are the precursors of hormone-like compounds, which is performed mainly by their fast growth rate and their potentiality to grow on various hydrophilic and hydrophobic substrates is derived from a fast-rating macroorganism on extremely large scale for the production essential to find single-cell protein and get into the realization with concern that microorganisms can compete on equal terms with cheap plant products provided that the scale of operation in fear of the depletion of oil resources and the global warming, present biotechnological research has concentrated on the commercial exploitation of microorganisms for the production of fuel. Therefore, there seems little reason why a process for the production of microbial oils and fats should not compete with conventional sources in an attempt to produce a better, more cost-effective commodity. Historically, the concept of microbial lipids being used as a supplementary source for oils and fats has attracted the maximum attention during the First and Second World Wars due to the disruption of trade and supply. Microbial sources of lipids and fatty acids has researched at an industrial level in many developed countries. Microbial lipids obtained from microorganisms oil produced was used for edible oil or even technological purposes all over the world. It's worth to basic therapeutic and note that microorganisms display more than the industrial production. The lipids within a cell may vary not only in amount but in type. Obtained process to be evaluated as efficient or what stage the industrial level microorganism is taken or fatty acids must be of high efficiency or high value. Though, like all living cells, the microorganisms, basically the yeast, fungi, bacteria, or even the microalgae, always contain lipids for the essential functions. In fact, single-cell oils can be employed as substitutes of expensive, not all microorganisms found in the can be considered as abundant sources containing oil high quantities. The microorganisms that have the ability to assimilate other neutral oils under certain specific conditions, or finally substitute around minimum 20% or more in the cell biomass are described as heterogeneous or oil-bearing or fatty. Specifically, in the last past years, there have been an increasing necessity for the discovery of non-conventional sources of oil and species, only around subsequently be converted into biodiesel or renewable diesel. We'll be interested in the several homogeneous microorganisms work as cell factories in order for single-cell oils to be synthesized on various carbon sources will be considered as potential realized by candidates for the production of lipids that will result by the generation of the second fungi generation lipid biofuels. Some microorganisms have a greater propensity to accumulate substantial amounts of oil in their cells, so the microorganisms which could accumulate lipids more than 20% of their biomass are called as oleaginous. The amount of lipids that can be found within the microbial cells, pellets or mycelia, may tremendously vary from less than 1% to over 80% of the dry age. The extent of lipids that can be produced in these microorganisms, a significant number of fungal species able to accumulate more than 25% of lipids have been already identified and characterized enormously among species that accumulated by fungi have higher alpha-station decrease than those are accumulated by the species. This is the principal reason for which a remarkable number of studies concerning lipids the most complex one by these microorganisms are mainly focused on the production of polyunsaturated fatty acids. Although the production cost of single-cell oil is currently higher than the traditional utilization, this is the principal reason for which oleaginous yeast strains are in general able to synthesize and accumulate species like up to 75 strains are considered as favorable oleaginous microorganisms and potential producers of species and strains with oleaginous character are Polyunsaturated fatty acids, Candida, Rhodopterin, and pharmaceutical Cryptococcus. For instance, in investigations have indicated that fatty acids have revealed or potassium salt, new genera, which is capable to produce containing non-medicinally important nucleic acids can have lipid quantities during growth of hydrophilic carbon sources in which the lipid production mechanism is promoted, such as coronation levels, employment of data-9 unsaturated cultivation in general, yeast lipids synthesized by the Senahol fatty acid biosynthesis mechanism is composed mainly of 16 carbon and 18 carbon saturated fatty acids. Salicylic acid constitutes the 55-25% of the total lipid, whereas stearic acid is generally a minor component. As we indicate previously, the yeast and fungal lipids occur not only as major constituents of membrane systems but also as cell wall components. As storage material in abundant and readily absorbed lipid bodies, whereas in some cases they can be met as extracellular products. In fact, storage lipids unable to integrate into phospholipid layers cluster to form the hydrophobic core of the lipid bodies or oil bodies. On the other hand, it is very important for the further development of economically viable and ecologically sound single-cell oil production. The biochemical studies suggest how microorganisms synthesize a definition for orogenic how they are able based on the position inside one of the key enzymes for we all know that in fact all living organisms must synthesize a minimum amount of lipids for the membrane and involved in lipid accumulation and function is monocytic. And the ATP required provides the homogeneous microorganisms acetylbinding for fatty acid biosynthesis as the non-homogeneous enzyme generates. Nevertheless, the presence of certain by which the acetylbinding production of single-cell oil is the backbone of the fatty acid. For lipid accumulation and biomass production for the microorganisms, there is a growth phase in which the microorganism adapts itself to the cultivation environment, and when the growth is limited, it will start another phase which is called as the lipogenic phase, where it accumulates the lipids in their cells. As far as the process of lipid accumulation from glucose enzymatically catabolized compounds and ethanol and low molecular weight organic acid is concerned, the so-called de novo lipid accumulation process originated from non-negligible number of yeast strains have been recorded to be biological requirement is the excess of growth on fat and carbon oil source and the deficiency of some other nutrients from the cultivation environment, which is generally nitrogen, that constitutes the limiting factor in growth medium. Under conditions of nitrogen initiation and the presence of excess carbon found in the form of several types of fungi, metabolic compounds to the general growth rate most rapidly and thus rate of carbon this results in the preferential channels of the are capable of presenting here we see the de novo and ex novo lipid in several cases. Growth of hydrophobic material is accompanied by accumulation of storage lipids by the implicated microorganism. This process is so called as the ex novo lipid accumulation process. The microorganisms that are able to present microbial growth on media based on 3-acylglycerols or other alkyl esters should obligatorily possess and express under the relevant culture conditions an active lipase system in order to break down the complex. For conclusion, we can say that there has been an almost twofold increase in the price of conventional plant commodity oil. If this situation continues, the orogenic microorganisms will soon constitute the major lipid source. Besides bioenergy aspects, in several other cases, oils and fats produced by allogenous yeast and fungi can present composition similarities with several high added value lipids, or they can contain polyunsaturated fatty acids of significant medical and nutritional value like gamma-linolenic acid. Thanks for your attention. Gamma-linolenic acid, arachidonic acid, etc. The orogenic yeasts and fungi in most cases are categorized as grass microorganisms, and due to their fast growth rate and potentiality to grow on several types of substrates, they are basically considered as the perfect cell factories for industrial, academic, and economic purposes.